Hello class, I'm creating this video to help you master some of the concepts we have studied in the previous lab and that are essential for you to remember as we continue with this exploration of the diversity of life. And you may recall that in the previous lab we were studying details about phylum Porifera, the sponges. We're also studying details about Phylum Cnidaria. And so as a brief review for the sponges, Phylum Porifera, remember that Porifera simply means covered with pores. And you may be asking, what pores do sponges have? And the answer is going to be two main types of pores. One of the pores is going to be the most obvious, and I'm going to switch here to my laser pointer function. Okay, so you can see the laser pointer moving around this opening of a sponge, which is going to be where water goes out. This type of opening, which is going to be large in size, is going to be known as an osculum. Sponges in general may have just one osculum or maybe two or three. What they will have numerous amount of will be these other smaller pores through which water goes in. Those are known as ostia. So ostia are the smaller pores. Osculum will be the larger pore where water is going out. Other details that I want you to remember about the sponges will be the types of cells. So we studied in lab this particular cell known as the choanocyte. This I call the signature cell of a sponge. If you're looking at a slide and you see these cells shaped like a vase, a flower vase, they have a color, and emerging from this color you have this flagellum. That is going to be known as a choanocyte, and they form layers inside of the body of the sponge, also lining the canals, and these flagella will be forcing water to move, usually in the direction out through the osculum. They also are responsible for collecting food through phagocytosis. This I call the signature cell of a sponge because only sponges, they are the only animals that have this type of a cell. And another important detail that I wanted you to remember about the sponges is that they are classified based on the materials that make up the endoskeleton. Yes, you heard correctly. This is an endoskeleton because the support for the sponge forms from inside of other layers of cells. Keep in mind that sponges are primitive animals, that they don't have real tissues, but they do have layers of cells. So here in this illustration, you can see the layer where the choanocytes are. Here you see another layer on the outside forming the exterior cover of the animal. These are called the pinacocytes. And then in between, there's going to be some amoeboid cells or amoebocytes. So that is going to be where the endoskeleton will be forming. These amoebocytes are going to be responsible for secreting the materials. In some sponges, those materials are going to be made up of calcium carbonate. And so here we have the class Calcarea. This is a subdivision of the Phylum Porifera. For the class Demospongiae, we are going to have sponges where the main component of the skeleton is going to be a protein known as spongin. And there's also going to be the hexactinellid sponges, also known as the glass sponges, because the material for the endoskeleton is going to be silica. And I said before, this is the same material that makes glass. In lab, one of the sponges you studied is this one known as Grantia. And Grantia is going to show the features about sponges we studied. For example, there's going to be this larger opening where water's moving out. This is called the osculum. There's also going to be a space inside of the sponge. This is going to be where water is collecting before it goes out. This is known as the spongocele. In addition to this, you'll see that the body of the sponge consists of several canals. There are some canals that are moving from the outside to the inside, and that is going to be known as an incurrent canal. If you look at this other illustration I'm providing, you can see where water goes into the incurrent canal through the smaller openings known as ostia. Just one opening is called ostium. Many openings, the plural will be ostia. And so here's one ostium, here's another ostium. Collectively, all the ostia allow for water to move into the body of the sponge. Once water enters into one of the incurrent canals, water will next move into internal canals that open into the sponge cavity or spongocele. These canals have a different name. The ones that are opening into the spongocele are known as radial canals. And you can see that name designated here in this illustration. Notice also in this cross-section of a sponge that there are illustrations for some of those amoeboid cells or amoebocytes, and there's also going to be the spicules, which are going to be, in this case, like sharp little needles that are providing the basis for the endoskeleton of the sponge. Moving over to the view, which you would have seen through the microscope, it is a lot harder to distinguish exactly where these amoeboid or cells or amoebocytes are, but you will see often this red layer of cells that is going to be the choanocyte layer. That is going to be one of the things you were asked, according to the lab instructions, to label. You were also asked to label the cavity inside, which is called the spongocele. You were also asked to label the osculum, and then these little openings where water moves into the incurrent canal, the little pores are called ostia. One is called ostium. And remember also that the radial canals will be those that are opening from the body of the sponge into the spongocele. So with regards to the sponges, I mean, those are the main details of the anatomy that you want to know, that you want to remember. For the phylum Cnidaria, this is going to include animals like sea jellies, sea anemones, corals, sea pens. There's a wide diversity of beautiful marine organisms that fit into this phylum Cnidaria. So what is the main criterion to fit into the phylum Cnidaria? They are also going to have a signature type of cell. I'm going to be changing to a pen because I want to write the name of this cell, and the cell is known as the cnidocyte. The first part of the word means stinger, like a stinger of a honeybee or a wasp that can be used for injecting a painful venom into the skin of another organism. And cyte simply means cell. So a cnidocyte is a stinging cell. And if you notice that the word Cnidaria makes reference to cnido, cnido, stingers. And so that's what these animals are. They're going to have these specialized cells known as cnidocytes to deliver a painful sting, which is going to be, in some cases, a paralyzing type of a venom to make it easier for them to catch prey. Sometimes the venom is powerful enough that would also kill the prey, not only paralyze it. And this is going to be their main means for obtaining food. The stingers, as you can imagine, can also be useful for defending the animal itself from potential enemies, like potential predators. So what we learn in lab is that these animals are going to have two main body types, and it kind of has to do on whether they are free drifters like jellyfish. We call them more appropriately sea jellies. Here's an example of a sea jelly. It's the body of a drifter. It's shaped like a bell. Here you see the idealized sketch of one of these bell-shaped animals. The official name is medusa. And so these medusas have tentacles surrounding the mouth region, because with these tentacles, they're going to be capturing small organisms, plankton, the larvae of other animals, so they can obtain the food, the nutrition to get energy from. The other type of cnidarian is going to be the one that, instead of drifting, lives attached to a surface below the surface of the ocean. So this is going to be a type of an animal that is attached to a rock, maybe a sandy substrate with rocks beneath. So these polyp bodies are going to be typical of anemones. Also, sea corals are going to have this type of a cylindrical body. And in terms of other characteristics, we learned in lab that cnidarians do have a gastrovascular cavity. These animals are a little more complex than the sponges because they do have tissues. And two main layers of tissues are recognized. On the outside, there's going to be a tissue forming the epidermis. And on the inside, there's going to be a tissue known as the gastrodermis, forming the gastrovascular cavity. Notice in this illustration, mouth surrounded by tentacles. Here's the mouth, mouth surrounded by tentacles. The tentacles bring food into the gastrovascular cavity, and their cells that are lining the gastrovascular cavity, cells of the gastrodermis, are going to be responsible for secreting materials like enzymes that will help digest the food. Remember that I said this is a closed type of a structure because it only has one opening. Food comes in one way, and whatever is not digested is going to be expelled out through the same opening. And I mentioned before that the mouth and the anus are the same opening. There's only one for food to come in and wastes to go out. These animals have a different type of skeleton. It's a hydrostatic skeleton because it is controlled by water pressure that can be regulated by the contraction of this gastrovascular cavity. So the animal can take up water or can release water, and that will be changing the water pressure and how they can maintain their shape. Examples of classes that one can list for the Phylum Cnidaria, we're going to have the hydrozoans, and we saw a couple of examples of hydrozoans in lab. One of those is Hydra, and another one is going to be the Obelia colonies. I'll tell you more about those in just a second. Another class is going to be Xiphozoa. This is going to be the true jellyfish. Anthozoa is going to be the anemones. Here you see an example of an anthozoan. The word literally means flowered animal, but we will get into that a little later in class. Here you see an example of a Xiphozoa, the moon jellies that you can appreciate if you go and take a ride on a ferry. If you look over the ferry towards the water, you will often see these beautiful, almost transparent jellyfish. And corals are also going to be examples of anthozoans. For now, I'm not going to say much about the cubozoans because it's something that we'll definitely explore later in class, not as part of the lab. So specimens you saw through the microscope that you capture pictures are going to include these two. Here's going to be Hydra. Notice how the body is cylindrical. This is a classic example of a polyp. These Hydra are going to have a mouth surrounded by tentacles. This mouth is also known at times, I'm gonna change to my pen function. This mouth is also known as a hypostome. So that's the mouth, the hypostome. Notice also that the animal is going to be reproducing at times asexually forming a bud. The bud is like a smaller version of the animal that continues to grow until it forms the features that you can typically recognize in an adult. For example, here you can see that the tentacles are beginning to grow. What would happen to these buds is once they reach a size where they can sustain themselves, they're going to self-clip, self-remove from the parent, and they're usually going to end up attaching right there close to where the parent is growing. And so they'll have their own place for attachment. I'm just going to draw here what one of these animals will look like growing after it detaches from the parent. So this little bud, what I'm saying is that it's going to detach and go and live right there as a neighbor to its parent. The one I see, or you see here to the right of Hydra, this is going to be known as an Obelia colony. And in an Obelia colony, what you will see is that there are two different types of polyps. Polyps that are useful for collecting food, such as this hydranth. This is a variation of a polyp that has tentacles. The tentacles, as you've learned from this review, they are going to have stinging cells useful for capturing prey. And so what they do is they capture the prey, they digest it, and then they share some of those nutrients with other members of the colony, of this polyp colony. Another type of polyp you see right here is going to be one that is used for reproduction. Reproductive polyps are known as gonangia. So gonangia means one, more than one will be known as gonangia. That'll be the plural version of this word, gonangium. And you typically don't see tentacles growing at the top of this gonangium type of a polyp. What you do see inside is little small round objects, and those are going to be juvenile medusa, like little tiny jellyfish that will eventually be released. Moving back to my pen function. So from in here, this little baby medusas are going to be released. I'm gonna make just a few to demonstrate what I'm talking about. So this will be like the bell-shaped bodies with the little tiny tentacles. These little medusas are going to be produced by the gonangium. And so the main mission of this gonangium is going to be reproduction. What happens later is that the little medusas go and reproduce, eventually making more of these polyps. It is a very special case of reproduction that uses both sexual reproduction and asexual reproduction. But again, that's going to be a concept that I'm going to explore with you in class at a later point. One of the details that I want you to remember as you prepare for submitting your lab write-up is that I do want you to make your own drawings. I don't want to see pictures pasted for specimens such as obelia, or specimens such as hydra you see over here. And the one I showed you previously, like your drawing for something like Grantia. I don't want you to include pictures. I want to see your actual drawings. One of the things that students say is like, oh, but wait a moment, I'm not an artist. I don't know how to draw. And what I want to tell you is that drawing these types of lab specimens does not require you to be an artist. What I'm looking for is like a technical representation of the types of cells or tissues or the organism you're seeing. And so drawing on a slide, on a PowerPoint slide, is real difficult. If you ever try to do that, you know what I'm talking about. But I will demonstrate to you how easy it is, what I'm expecting from you. So for something like a hydra, I'm going to make a very long cylindrical body. There's going to be a disc where this animal attaches to, like the surface of the ocean. So here's going to be the pedal disc. By pedal, I mean like foot, P-E-D-A-L. And then this hydra maybe has like a little polyp that is growing. And so I'm gonna make the little tentacles of the polyp. The little hydra bud. And so again, this is going to be my little hydra bud. And notice that I'm making a representation that is more than just a scribble. It's actually showing that this is an animal with a cylindrical body. It is going to have a mouth. This is going to be where the mouth is. And there's going to be tentacles. They are going to be really long. Some of them are going to be making kind of like, you know, curves depending on, you know, what the tentacle was doing when this animal was pressed into the slide. And so I'm taking the time to draw some tentacles, which is something that you can do much more easily if you are doing it on the space provided in your lab worksheets. And so by now you get the idea of what I'm trying to do. So this is an example of hydra, and you're going to be labeling that. This is going to be the bud. This is going to be the mouth. These are going to be tentacles. And you're going to be labeling those things you're drawing. What I do not want to see in the lab notebooks is the lame excuse like, oh, I don't know how to draw, I'm not an artist. And then people try to pass by me something like a hydra that looks like this. And this is what they're submitting. It's like, absolutely not acceptable. I don't know what that is really, this over here. So this, no. This, yes, absolutely. I want you to do that. And you can do the same thing with something like Obelia. So it's like a colony, and the little polyps are living kind of like on a tree. And so I'm gonna make some of the branches of the tree. I'm not looking at one, I'm just drawing one from like memory. And so I'm going to include here maybe one of those polyps that specialize on reproduction. And so remember that these polyps that use reproduction or are used for reproduction do not have tentacles, but they do have inside the little medusa buds. And so I'm taking the time to draw some little tiny circles in here representing the little medusa buds. And then I can label this. So this is going to be my gonangium. And these are little medusa buds. And maybe over here on this side, I'm gonna be drawing something more like a hydrans with the many, many tentacles these hydrants possess. And so I have my two types of main polyps, and you can add, you know, a few more just to make your drawing complete. And so this is what I mean by you can draw things, and you don't have to be an artist, but you can show the technicalities. Here's a polyp, no tentacles. Here's a polyp, yes, it does have tentacles. The ones with tentacles are used for the mission, the contribution is to do feeding. That's the hydrant. This one over here doesn't have tentacles, but it does have little juvenile medusas inside. So the function, the contribution to the colony is going to be participating in reproduction. And so make your drawings, make them the best you can. You're going to learn a lot from making the sketch, from labeling the sketches you're making. And you can expect that on the quiz, there's going to be illustrations for you to recognize these kinds of structures I've labeled here, and also the type of specimen you're looking at. So listen carefully. Yes, on the quiz, I'm going to have like an example of a hydra, an example of Anovelia, Grantia, the sponge, and your job is going to be recognized. Yeah, specimen A, that's Ovelia. Specimen B, that's Grantia. So prepare adequately for the quiz, and I hope that you find this review helpful. Goodbye.